process of production of male and female gametes in human, let us proceed further. In today's class, we shall know about how fertilization takes place in human, the process of fertilization. Let's start. So here, as you can see, there is a diagram of sperm, human egg, and the reproductive structure. So in this class, we shall come to know the process of fertilization right from when sperms are deposited in the vaginal tract by copulation, that is by sexual intercourse. When sperms are deposited in the vagina, from there it goes all the way to the oviduct and fertilizes. Fertilizes means unites with the egg cell. So from here to here, this is the whole process that we shall come to know in this video, and that is what the process of fertilization is all about. Now, before going into fertilization, let's recap once again the structure of human sperm and human egg. So here, as you can see, this is the structure of human sperm. Human sperm, just recall, it is 4.5 micron in length, 2.5 micron in diameter. So this is human sperm. It consists of three parts: head, middle piece, and tail. Three parts: head, middle piece, and tail, and they have their specific function. Sperm, mind you, it has the only function to travel and fertilize the egg cell. That is the function. So before fertilization, it has to travel a long distance, and for that reason it has been made into this special structure, and it is provided with special features which will aid in its travel. So the first is head portion, which has acrosome and nucleus. So nucleus is the thing which it has to deliver to the egg cell. Acrosome is a body, is an organelle which is developed from Golgi body, and it contains a number of enzymes which will assist in this process. And finally we have middle piece, which has mitochondria. Middle piece has got mitochondria, because mitochondria is the thing which will provide strength, which will provide the necessary force which will be required for travel. So mitochondria, middle piece has mitochondria. And finally we have tail, a big long tail which is lashing in its nature. It lashes so that the sperm can travel forward. So tail helps in movement. So this is the structure of a sperm. Now let us talk about the egg cell. Egg cell, this is known as secondary oocyte. It is not yet egg cell. Keep this in mind. It is secondary oocyte because the nucleus has not finished its process of meiosis. It is still held up in meiosis 1. It has released only one polar body. One more polar body needs to be released. So it is secondary oocyte. Now, what the structure looks like, let us just try to recap. This is a secondary oocyte, this is the nucleus. Then we have plasma membrane, this is the plasma membrane. Over the plasma membrane, there is a layer which is known as zona pellucida. This layer is a thick layer, it is known as zona pellucida. It was there when this was in Graafian follicle. Zona pellucida was there also. Zona pellucida. And over the zona pellucida, there is one more layer of granulosa cell. Remember granulosa cell of the Graafian follicle, granulosa cell. Now, when the secondary oocyte ruptures, it carries with it some amount of granulosa cell with it. And this is known as corona radiata because it tends to radiate in all directions. See the spikes? This is known as corona radiata because it radiates like spikes. So corona radiata is nothing but granulosa cell. So this is the structure of secondary oocyte. This is the structure of male sperm. Now, what is the size of secondary oocyte? Keep this in mind. Secondary oocyte is 100 times bigger than the sperm. Its size is 0.1 millimeter. 0.1 millimeter. So sometimes difference might also be out between the sperm and the secondary oocyte. So you can very well see there are hundreds of difference between them, but to keep 2-3 points in mind, the size is a major difference. This is 4.5 micron and this is 0.1 millimeter. This has got no extra structure in it, no food stuff, nothing. It has got just mitochondria and acrosome and nucleus. It has got a lot of food stuff because it has to sustain itself. So it has got yolk, it has got other organelle as well, and on top of that it has got a lot of layers around it. So these are the structures of these two units which will finally unite. Now let us come to this diagram. It is the reproductive structure of female. Here is the process of reproduction, the fertilization will take place. So let's start from this point. When the sperms are deposited on the vaginal tract, that is by copulation, sexual intercourse, where sperms are deposited at one ejaculation, one copulation, the amount of sperm, the number of sperm that is deposited is 100 million. 100 million, all MCQ questions. 100 million sperms are deposited at one ejaculation. Now, sperm, it will always be traveling in a medium, in a dense medium. So, the travel is going to be very difficult for it. Just imagine like you are made to swim in a very dense pool. So, how will it be? So, you will need lot of features to yourself which will aid you in that process. So, similarly, sperm has those structures which will aid to move in a dense medium. So, sperm will travel, first of all, lot of sperm will disintegrate at this very point. Then, the stronger ones, they will go up the cervix and travel all the way from the uterus, both the side, to the oviduct. Now, oviduct and fallopian tube are the same thing. Fallopian tube are the same thing. We continue saying fallopian tube when we discuss the production of gametes, but when it comes to fertilization, fallopian tube, we replace the word with oviduct. Oviduct, okay, the same thing. So, fertilization will be taking place in the oviduct. Now, the sperm has to travel all the way through uterus and meet the egg somewhere in the fallopian tube, that is the ampulla region. It will meet the egg here. Now, there are some features which needs to be understood in this context. First of all, the sperms which will be going forward, they will have to make themselves even more stronger. Okay, and this process is known as capacitation. Capacitation, because just remember the size. The size of the sperm is very small, 4.5 micron, and this distance, if you remember the distance, it is somewhere around 10 centimeter. Okay, it has to travel around about 8 to 10 centimeter. The uterus itself is 7 centimeter in length. It will be like you are swimming from here right up to Gujarat. Just imagine, you will be swimming from here to Gujarat in a dense medium. So definitely you will be needing a lot of features which will help you in this. By yourself you will not be able to do that. So that is what the process of capacitation does. It provides extra features, extra gadgets or extra strength to the sperm so that it can carry the movement. So capacitation is basically bringing changes in the sperm itself. The tail will be all the more active. It will lash even more so that the sperm travels properly, and the sperm will become more stronger. That is capacitation. On top of capacitation, there are some other things which help sperm in this movement, and that is muscle contraction, because the muscles of uterus, they will be contracting, and that helps the sperm to move forward. And then there is one hormone which is known as prostaglandin. Remember that accessory sex organ in man, prostate gland. Prostate gland had given this prostaglandin to the sperm, and this is where it will help the sperm. It will make the path little more dilute. It's very dense. It will cut that denseness. Okay, it will make it little lighter so that the sperm can move. So prostaglandin. So these three things help the sperm in the movement. The distance it takes. The time it takes to cover this distance is 45 minutes. So just imagine, even if we go in flight from here to Gujarat, it will take us more than 2 hours. So just imagine the speed that the sperm has to cover this distance. It is even more than the flight. Okay, and it's doing all by itself. It does not have any other vehicle with which it is going. So it's like we are swimming all the way. So that's what. So it will come to the oviduct and finally it will meet. Now there are other features which we need to talk about before this meeting takes place. Let's do that now. After facing all the hardships, few of the sperms will finally reach the egg cell. Then they will try to penetrate the egg cell, but the fittest one of them all will be able to do so. Keep in mind, the acrozote of the sperm has got few enzymes. We had spoken about this. Now is the time to see what those enzymes are and what are their functions. Let's do that. So, sperm has number one enzyme which is known as hyaluronidase. Hyaluronidase. Egg cell has corona radiata which has hyaluronic acid. So the fittest sperm will have the perfect hyaluronidase to penetrate or to dissolve this hyaluronic acid. So this layer will go away because it will be dissolved by hyaluronidase. So first layer gone. Now then comes yet another enzyme in sperm which is antifertilizin. Antifertilizin. And the cytoplasm of egg cell has got fertilizin enzyme. Now, antifertilizin and fertilizin, they will unite and this is species specific. That means a monkey's sperm will not be able to fertilize a human egg. Species specific. Only monkey, homo sapien, cat, dog, that way. It is species specific. So antifertilizin is in sperm and fertilizin is in the egg cell. So they will unite, and with this union there will be a path created. There will be a fertilization path created. This path along which the male nucleus will travel, this is known as fertilization path. Fertilization path. This fertilization path is created. Fertilization path means it is cytoplasm, isn't it? Cytoplasm is very dense. Now in that dense medium, a shallow portion will be created because of these two enzymes, antifertilizin and fertilizin. They will react with each other and they will nullify the effect of each other, creating a path. Along this path, the male nucleus, which is not known as nucleus as yet, it is known as pronucleus. The male pronucleus will go along that path to unite with the female pronucleus. Now let us just talk about pronucleuses. What are they? Remember, when it was in the sperm, when the nucleus was in the sperm, at that time it was very much constricted. Okay, so as it enters the cytoplasm of the egg cell, it expands a little. The chromosomes, they spread out a little. This is known as male pronucleus. When male pronucleus is being formed, at that time the nucleus of the egg will also get the required stimulus, and it will wake up from its meiosis one stage, and it will also finish its meiosis. That means it will undergo meiosis 2 and take out one more polar body. So now this nucleus will become female pronucleus, and this stage it has become a true ovum. And before this it was just secondary oocyte. So most of the egg cells, they vanish in secondary oocyte stage only. They don't become ovum. They become ovum only when a sperm comes and unites with it. So at that time, that moment it becomes ovum. Now there is one more thing left. When the sperm was entering, just near the cytoplasm of the egg cell, there are few granules which are known as cortical granules. Cortical granules. Now, what is the function of a cortical granule? When the sperm will penetrate the egg cell, which will try to enter the cytoplasm, there will be a reaction in this cortical granule, and they will all be released. That means they are in the vesicles. Those vesicles will break, and they will unite with the plasma membrane of the egg cell, and they will form a very thick fertilization membrane. So this happens after sperm is penetrating into the egg cell, and then the cortical granules, they break and unite with the the plasma membrane and makes it a thick membrane which is known as fertilization membrane, so that other sperms cannot penetrate the egg cell. The function of fertilization membrane is to stop the other sperms from entering the secondary oocyte. Only the head portion of the fittest sperm will enter the oocyte, and even the tail portion will remain out, and after that cortical granules will be released and fertilization membrane will be created. So we have male pronucleus, we have female pronucleus, there is a fertilization path already made, and now union takes place. Union takes place between these two nuclei, and a diploid nucleus is formed, a diploid organism, a cell, diploid cell is created, which is the first cell of this entity. This is known as zygote. It has 46 chromosomes. When zygote is formed, when the union happens between these two nuclei, that time the female is said to be pregnant. This is the onset of pregnancy, when fertilization has taken place. The egg cell remains viable only for 12 to 24 hours after ovulation has been done, and the sperm remains competent only for 48 to 72 hours. That is their lifespan. Within this time, fertilization should take place if it has to happen. So that is the time period, and this is how fertilization takes place. So we have come to the end of this video. This video tried to explain to you how fertilization happens in case of human beings. After this, we shall be discussing how the development of embryo happens up till blastocyst stage. That will happen in the next video. So please keep following my video, and thank you for watching.